Okay, so today's class is going to look at leaf structure. And of course, this is a leaf, and we probably know it from CSEC days. So we have an outer waxy layer, which is called the cuticle. And the cuticle covers upper epidermis. This calls it adaxial epidermis. It's an adaxial epidermis, or the upper epidermis, followed by the palisade cells, or called the palisade mesophyll. And of course, mesophyll means middle leaf. Then we have the spongy mesophyll. The point about the spongy mesophyll is you have lots of air spaces, and this is for gaseous exchange. We also have a vascular bundle, which contains the stick-walled cells, which are lignified, which form the xylem. And we have thinner walled cells, which form the phloem. And surrounding it, we have the bundle sheath cells. The bundle sheath are important because they actively pump sugars into the phloem. And remember, the phloem is important for the transport of organic substances to the rest of the plant. Surrounding the vascular bundle, we have parenchyma cells. Parenchyma is basically packaging, although sometimes the cells can form collenchyma or sclerenchyma, and we'll look at that in a bit later. And finally, we have the lower epidermis, or as this thing calls the abaxial epidermis. And you have a stoma here. Stoma means mouth, and it allows air to get into this airspace. And all the air spaces are interconnected. And that's important. The air spaces need to be interconnected so that gaseous exchange can occur. So what we're going to be looking at is photosynthesis and ATP synthesis. And we're not going to get into too much detail on ATP synthesis in this presentation. That will occur later. But what we look at is students should be able to relate the structure of a dicotyledonous leaf, a palisade cell, and a chloroplast to their roles in the process of photosynthesis. And this requires annotated diagrams. And we're going to do a drawing, in fact that should say drawing, of a prepared slide of a transverse section of a dicotyledonous leaf. But again, this will be for a next class. But we'll briefly move on to the next part. Explain the process of photophosphorylation. Well, we actually won't get onto this. We'll just look at the structure of ATP, and that will be it for this class. So, looking at the structure of a leaf, remember we need to know the structure of a leaf, and it needs to be annotated. If I have an epidermis, which is the outermost layer of the cells, and cuticle on the surface keeps moisture inside. So the cuticle prevents water from evaporating. The cuticle also contains no chloroplasts, so it doesn't photosynthesize. And that's important. Really, there's no air spaces here. So the cells couldn't get enough carbon dioxide anyway. So therefore, there's no chloroplast so that light can penetrate through it. Then we have the palisade cells, which are long and thin, and they're closely packed, although there are air spaces. And this is important, as I said, for gaseous exchange. And being long and thin allows maximum packaging of chloroplasts. Remember that the palisade cell, this palisade layer, is the most active layer for photosynthesis. Then we have the spongy layer. These are loosely packed, and this is really for gaseous exchange. And then we have the guard cells. And the guard cells regulate the opening and closing of the stomata. So in general, they open during the daytime and close at night time. They're open during the daytime so that gaseous exchange can occur and carbon dioxide can enter. And they close at night time to prevent water loss. Because one of the drawbacks of the structure of a leaf is it allows water loss, and the leaf loses a lot of water. And what did I say here? The cuticle is transparent to allow light to pass through it. Palisade cells are cylindrical in shape, and this allows maximum packaging of chloroplasts. Cells in upper epidermis lack chloroplasts so light can pass through them. Spongy layer contains many air spaces, and this maximizes gaseous exchange. And I said some leaves also contain parenchyma cells surrounding the vascular bundle. And these, as I said earlier, can be modified into collenchyma, where the cell wall is a bit thicker, and this gives a more rigid support. It gives mechanical support for the leaf. And sometimes it can actually become sclerenchyma, where the cells die, and the cell walls become lignified. And the vascular bundle also gives mechanical support to the leaf and provides it with water so that it can remain fully turgid. And the leaf needs to be turgid so that photosynthesis can occur. If the leaf wilts, it can't photosynthesize. Okay. And the bundle sheath cells actively pump sugars into sieve tubes. So again, we can look at the structure of a leaf here. Again, another diagram showing the structure of the leaf. This one is slightly easier, I think, so that if you're asked to draw the structure of a leaf, you could probably draw something like this. And remember, waxy cuticle prevents water loss, but allows light to pass through. Then we have the upper epidermis, which is transparent, which allows light to pass through it. And it's also protective and prevents the entry of pathogens. Followed by the palisade cell. These are tall and thin and closely packed together and contain a large number of chloroplasts. And it allows for maximum absorption of light. And you have the spongy mesophyll. Lots of air spaces for gaseous exchange. And the lower epidermis has pores called stomata to allow carbon dioxide to enter into the leaf. And you have guard cells which can open and close. So also the lower epidermis cells do not contain chloroplasts either. And then you have a xylem, which brings water to the leaf, and that keeps the leaf fully turgid. So the photosynthesis can occur. And phloem, which takes sugars away from the leaf to where it might be needed. And then the palisade cells are adapted to the function of photosynthesis. They have a continuous layer to absorb sunlight, and many chloroplasts around the edges. Notice you have a vacuole in the center. So the chloroplasts are pushed to the edges of the cell. And the function really is to absorb as much sunlight as possible. And here we have a structure of a chloroplast. You might remember this from Unit 1, where we have a double membrane. And this also gives evidence for the endosymbiosis origin for the chloroplast. But the double membrane is important in, well not really in photosynthesis, more important in the mitochondrion, but it's evidence for the endosymbiosis. And the stroma has an appropriate enzymes and suitable pH for the Calvin cycle. The Calvin cycle of the light-independent stage of photosynthesis. This is where sugars are made, basically. And the thylakoid, well, the thylakoid contains a stack of sacs. Well, the granum contains a stack of flattened sacs. And the fact that these are flattened is important, so that part of the chlorophyll molecule can rest on the top of each sac, and another part of it can burrow into the membrane itself. So a thylakoid has certain the thylakoid sac contains photosystems and enzymes and so on important for photophosphorylation, which we'll look at in a minute. And then you have a lamella, which connects granum together. And each granum really is a sac of thylakoids. So we're going to look at the structure of a chlorophyll molecule. And a chlorophyll molecule looks like this. You have a phytol tail and a porphyrin ring. The porphyrin ring is totally flat, and that's because these carbon atoms are all sp2 hybridized. You might remember sp2 hybridization from chemistry last year. And when we have sp2 hybridization, the molecule is the atom or the bonds around the carbon atom are flat. And because all these carbon atoms are sp2 hybridized, the porphyrin ring is totally flat. The phytol tail is not. So the porphyrin ring lies on the top of the granum sac, and the phytol tail dissolves into the membrane. And you can see that here. The long phytol tail dissolves in the thylakoid membrane, and the flat porphyrin ring rests on the thylakoid membrane. And it's the porphyrin ring which absorbs sunlight. And when it does so, the electrons in the molecule get excited. It's something, again, we'll talk about in a short time. And several chlorophyll molecules associate together to form a a protein molecule from a photosystem. And this is where light absorption occurs. And the thylakoid membrane can hold a large number of photosystems. So if we look at the structure here, each of these round circles really is a photosystem which contains many porphyrin rings and other associated pigments which absorb light for photosynthesis. So have chlorophylls and other pigments, and we call these PAR, photosynthetically active. They absorb PAR, photosynthetically active radiation. So chlorophylls are embedded in the thylakoid membrane, and they're arranged into a photosystem, which is a collection of molecules. So following that, we're going to look at the structure of a leaf. And in a later class, we're going to draw this leaf. And we're going to go over the structure of a leaf. But that is for a next class. And we're going to draw a plan diagram. As I said, this is something we're going to go on to in the next class. And then after that, we're going to look at the structure of a chloroplast. And I said a chloroplast looks like this, where you have a granum, a sac of thylakoids, have a lamellae which connects the grana together, and each granum is a collection of thylakoids. And you should be able to identify the different structures here. You can see lots of lamellae. You can see these darkened regions, which are the grana, and each granum consists of, well, as I said, each granum consists of a collection of thylakoids. And from that, you should be able to identify A, B, C, and D. And you're supposed to do that for yourselves. And again here, you can see an outer and inner membrane. You can see the granum, the lamellae, the stroma, quite often you see starch grains, which you can't see here. And then after that, we just mentioned the structure of ATP. So this is a molecule of ATP. You have an adenosine molecule. And of course, adenosine contains what's it called? Deoxyribose together with adenine, and then 3 phosphate molecules. The final phosphate molecule is highly energetic, and it is when it is hydrolyzed, it releases energy and forms adenosine diphosphate. But of course, this reaction is easily reversed, although it requires energy to reform ATP. And this is the energy currency that all biological molecules use. So if organisms want to spend energy or use it up, they've got to convert that energy into ATP. And we'll look at this in more detail in the subsequent class. But we can see how ATP is used. Mechanical, uh, such as muscle sliding, filament, cilia and flagella, propulsion, chromosome movement in mitosis and meiosis, all of these require uh, ATP. And ATP is broken down into ADP and PI, that is inorganic phosphate. Active transport also requires ATP. And then chemical reactions, anabolic reactions, uh, endothermic reactions, all require ATP. We'll probably can look at those in more detail in a subsequent class. So this basically covers what we wanted to look at in today's class. So that will do.